potency. It was originally developed as an antiviral against other kinds of viruses and has been tested in several animals where it has shown some effectiveness. But we think, and in our hands, we have found that biochemically it is quite potent, but not. Oh, excuse me. Okay. Okay. So I was saying that the potency is quite good, but it really needs to have improved potency to be a really good drug for humans. And it also needs to have better pharmacokinetic properties to be best suited for human use, meaning that it can be better absorbed orally as well as being resistant to metabolic degradation. For those of you who are familiar with the chemistry, this is what we call a peptidomimetic. That is, the chemistry is closely based on peptides, and this is based on a dipeptide. So what our group is focusing on now is looking at variants of GC376 and trying to modify it for improved potency and pharmacokinetics. Okay, now let's go on to the AI part of the talk. So let me give you start with a broad background, and then we will narrow into applications to drug design. So what do we mean by when we say AI or machine learning or deep learning? These are terms that you hear a lot and are hot buzzwords. They are sometimes used interchangeably, but actually their meanings are quite different. So artificial intelligence is a very broad term that includes basically any kind of algorithms that give machines this almost human-like intelligence for processing. That can be that's defined very broadly. These days, almost all of the most important work is in the area of machine learning. So machine learning is basically using statistical models and large data sets to train algorithms to be able to make predictions or to do what we will describe later as generative algorithms, that is, to come up with new ideas. Deep learning, this is the hottest area, one of the hottest areas of machine learning. Deep learning means using large neural networks, much larger than those that have been used in the past, and also using extremely large data sets to get very impressive results. This method has only been useful very recently, let's say within the last 15 years or so, because it required new algorithms and it required new levels of processing power and data storage that we did not have in the past. So first, let me just give you a very brief introduction to what is a neural network. A neural network is very simple. Here, you start with a node in this circle, and the node can receive several input signals. Each of these input signals is associated with a weight value. And the node applies a simple function, and it gives an output value. Now, these nodes are connected together in what is called a neural network, so that one layer of nodes takes the output from the previous layer of nodes and then performs that same kind of function and then feeds its output into another layer. This gets complex in practice, as now people are experimenting with deep neural networks, which may have 50 or more layers of nodes. Now, typically in chemistry and in physics, because, and I'll come back to this point later, because we generally do not have the massive amounts of data to train these neural networks, we tend to use simpler neural networks instead of very deep ones. So typically these days in chemistry and physics, we might use neural networks that have on the order of 3 to 5 to 7 layers, unlike the ones used for image recognition or image classification. Now, this is all very complicated and technical. What we're more interested in is what is the point and what is the power of using large neural networks. And really what it comes down to is that it has been shown that by using neural networks, one can approximate any kind of function. So this can be a function that, for example, distinguishes between pictures of dogs versus pictures of cats. Now, for us in drug discovery, we are interested in a function that can predict the activity of a molecule given the molecular structure. Now, the power of this approximation increases as your network complexity increases and as the amount of training data that you have for this network increases. So in many ways, what we're talking about when we say machine learning is not really that new. It really takes a lot of concepts that for a chemist, or especially a physical chemist, are already familiar. For example, using probability and statistics, the concept of global versus local optimization. For example, in chemistry, we're often interested in global versus local optimization of potential energy functions and landscapes. Cross-validation. Cross-validation is a statistical technique that can help us determine whether our model is overfitting our data. So just like in the simplest level, what we're doing with the neural networks and machine learning is that it's similar to fitting a curve. You can fit a curve with a linear function, or you can fit a curve with a very highly complex function. But if you use too many parameters to fit your curve, you can get a very nice fit, but it has low information value, meaning it has low predictive value because it's just an arbitrary fit to your curve. So these are kind of issues that are familiar to us as structural and physical chemists. So I say that because I want to encourage those of you with a background in mathematics or probability and statistics or any of the physical sciences to not be afraid of learning the latest methods in machine learning. Okay, so what is special about applying machine learning to scientific issues? So as I mentioned before, this is something like curve fitting. So for us to have good fitting, we need to have large, and I didn't state it here, but we want to have large, but also high quality data. By what do we mean by large? Ideally, we would have thousands of data points, even millions. And we can, however, this is a big challenge because in most of the cases, in experimental science, there are no data sets that have such of such size. So especially if you think about in an academic setting, a person might work for a year on a project and might have only 50 or 100, a few hundred data points into their experiment. So we can only do this, apply machine learning to a few special cases, it seems. So the data for applying machine learning in science is scarce, and it's expensive and slow to come by. Meaning that if we want to add more data points in the area of drug discovery, it means that a chemist might have to synthesize a new molecule, and then someone has to perform assays of this new molecule. So that means you could require several days to several weeks and many thousands of dollars just to acquire a few more data points. So let's step back a little. So why do we care about the data? Well, this comes from a paper that was published by Google Research several years ago called The Unreasonable Effectiveness of Data. And here they were looking at image classification algorithms. And what they saw was that by using deep learning techniques, that as you increase the number of examples, and you see here on the x-axis that this is in the millions, that the accuracy and the power of the algorithm continues to increase, which means that it does not plateau and level out. That as you continue to add data, the machine learning algorithm can continue to learn and improve seemingly without limit. So that's very impressive. Now, how do we apply this to drug discovery? Well, there are several areas where we have very rapidly growing data sets. So for example, protein crystal structures. Now we have several 100,000 crystal structures available. So that helps a lot. If we look at something like membrane protein crystal structures, we see that that this has also increased drastically over the years, and that there are now over 1,000 membrane protein crystal structures. So that's important because many of the targets of drugs are membrane proteins. And also, as the new technologies like cryo-electron microscopy advance, that has rapidly accelerated our ability to determine new crystal structures. And we expect this to continue to increase exponentially in the years ahead. So we have increasing levels of data that we did not have just 5 or 10 years ago. One of my colleagues have done very interesting work applying quantum mechanical methods with deep learning. And what they have shown is that they can train deep learning methods with data from high accuracy quantum computing methods to develop machine learning methods that can estimate these very computationally expensive quantum chemistry methods, but with 100 to 1,000-fold acceleration. And so what they do is they perform enormous sets of data sets, enormous numbers of these high-level quantum chemistry calculations, and then use machine learning to approximate it. And this is one of the most exciting developments, I think, in chemistry related to drug design. Okay, so we come back. So I want to summarize this part of the talk and say that a fundamental challenge is how to work with smaller data sets in drug discovery and chemistry. And what we need to do is, if we can restrict our scope of our problem, then we don't need as much data. So what, for example, if Google is not interested in classifying every kind of image, but just classifying pictures of dogs versus pictures of cats, then they don't need a million data points to do that. They can do this with far less data. And the same thing applies to our work. What the quantum chemistry work has demonstrated is that this training data does not necessarily need to come from experiments. We can use, if we use good calculations or simulations, that might be a good substitute for experimental training data. And just like any kind of data science problem, the quality, the type, and representation of the data is critical. And I'll show you how this issue comes up in our research. Okay, so the AI applications to drug discovery, I'm going to focus on the areas where we're working on, which is one, predicting drug activity from chemical structure. So this is a fairly better developed area of research. And we and others have shown that machine learning methods can usually outperform traditional computational methods for performing docking and virtual screening, as well as for QSAR, quantitative structure activity relationship types of calculations. And then another thing that our group is doing is that we're using something called generative machine learning to do molecular design. So this field of research is a little bit more bleeding edge, and the results are perhaps not as directly useful at this point, but we hope to get there very soon. Okay. So the first aspect, calculating protein-drug interactions. So this is an area where we and others have shown that using machine learning can significantly outperform conventional methods. So what we do is we extract the chemical features. By features, we just mean data from models, structural models of protein drug complexes. Now, this data can be any form of data. It can be something as simple as molecular weight, or it can be something more complicated, such as quantum chemical properties or structural coordinates. We train a machine learning algorithm on data sets of correct and incorrect docking orientations. That way we can train the model to distinguish between correct and incorrect. And of course, not just to distinguish, but to make this quantitative. If this works, what this can do is that this will help identify the most significant features. So this goes back to step one. So now we can interpret the results of the machine learning, and we can say what features are the most important. So they can be something that we already know. For example, that there should be electrostatic complementarity, or perhaps it will identify new features that are not so obvious to humans. In that way, we're not just creating an algorithm, but we're creating new chemical knowledge for humans as well. And then what we end up with is something that is called a scoring function. And a scoring function is something that you apply to a structural model of the protein drug target with the drug to make a quantitative prediction of the activity or binding constant. So let me give you a little bit of background and introduction for those of you who are not used to this field. So this is basically the principles behind what we call structure-based drug design. So we have a protein, maybe we know the binding site, of a drug. So here's a potential drug molecule. So we use computer algorithms to fit this ligand into the binding site. And we can use different kinds of scoring functions to predict what conformation the ligand takes when it binds into the site. And we call this the pose of the molecule in the binding site. So that's one problem, is that we want to predict the pose, and then we want to be able to predict the score, which represents how well the ligand binds into that site. So one of the best areas, so I often get asked by students, how can I learn about the latest techniques in computational drug discovery or virtual screening? So one place I highly recommend to learn about the latest work is to follow the blinded competitions that we have So, for example, the most prominent one is called the Drug Design Data Resource Challenge, D3R. And at the latest competition, we were asked to we were given a number of inhibitor molecules and to predict how well they would bind and what was their correct binding pose to a Alzheimer's disease drug target, actually in this case, a protease called BACE1. Now, a drug company has already solved the crystal structure of all of these inhibitors with BACE1, and they have already determined the binding constants, but they're holding that secret, and then the participants will try to make their predictions, and at the end, they see which team made the most accurate predictions. And by using relatively simple machine learning neural network methods, our team was able to place 4th for ranking activity, drug binding activity. And this just shows you what the competition involves. So here, for example, this was to rank ligands, 20 unknown ligands against a secret crystal structure. And here we show our predictions versus the known crystal structure. And you can see the match is quite good. The conventional measure of success is that there'd be a 2.0 angstrom root mean square deviation in the atoms. And so we did quite better than that. But although this was quite good to us, other groups did much better than us. So that's good. That's good for the field. That means that the field is advancing to the point where we can often accurately predict the pose. What is harder is to predict the binding affinity. And just to demonstrate the molecules that we were trying to do predictions with, these are complicated macrocycles with a lot of flexibility. So the way we were successful were to combine the conventional structure-based features. So these are features that are based on the 3D coordinates of the crystal structures or models with simple ligand-based features. So these are the kind of properties that we call 1D or 2D chemical properties, often used in QSAR types of research. Simple properties such as how many hydrogen bond donors and acceptors there are, what is the molecular weight, what are the patterns of electrostatics. And by combining these 1D, 2D, and 3D features using a relatively shallow deep learning architecture, we're able to achieve one of the best results in our predictions. So this is now something that is routinely done by leading research groups and by research groups at leading pharmaceutical companies. What I want to move on now is to talk about something that is a little bit more bleeding edge, which is the concept of using generative machine learning to come up with new molecules. So more broadly, this problem is called the challenge of inverse design. So we are starting with a functional space, meaning that we have some desired properties. For example, we want our molecule to be drug-like. We want it to be effective, and we want it to be soluble and non-toxic, et cetera, et cetera. So you can use any kind of property that you can define in a quantitative manner. And how do we map these desired features into chemical space? That is, how do we go from function to real molecules that we can synthesize and test? So this is inverse design. So the most famous case of using generative AI is coming up with photos of humans. So what this type of algorithm does is you train the computer algorithm with many photos of humans, and then you ask the algorithm, okay, now you come up with some examples of human faces. And all of these examples here, all of the photos shown on this page are computer generated. These do not correspond to real people, but this is what the computer algorithm has learned that what human faces look like. And if you look closely, you can see some artifacts and errors that throw it off. But for the most part, this is very impressive, at least from a human perspective, that the computer truly has seemed to learn the essential aspects of what a human face looks like, without being told specifically, for example, that there are two eyes that are positioned here and there, etc. So we want to take that same kind of approach and apply it to chemistry. Now, it's hard to implement in the computer all of the rules of chemistry. That's too complex. Just like you can't give a computer all of the rules for a human face. So what we do instead is that we feed the algorithm training sets of tens of thousands of molecules and have the algorithm infer what are the properties of the training set. So, and we try to give the computer some help. So we tell it, for example, that these kinds of molecules, they're like points that are connected in space, for example. And that there are some simple rules that tell you how atoms in a molecule can be connected together. And so we're doing this, and we want to see how well a computer can come up with new molecules that can fulfill desired functional goals. So this is a hard challenge. This is work that we've been collaborating with for a long time with Professor Alana Spuruguzik, who's at the University of Toronto. So his lab is one of the world leaders in this area, and we are working with him to apply this to drug chemistry. So one of the challenges of working with chemistry is that the space and diversity of molecules is enormous, meaning that if you are to think about how many different kinds of molecules one could make using, let's say, 15 or 20 atoms, so not a very large molecule, a medium-sized molecule of relevance to drug chemistry, it turns out that this space is enormous, much larger than, for example, 10 to the 20th size. This is something on the order where it's large enough that there is no way we can computationally examine the vastness of this space, much less sample it experimentally. So how do we do but the good thing about diversity is that this is not only a challenge, this is one of the wonderful things about chemistry, is that we can combine relatively simple atoms in almost an infinite number of combinations to get molecules that have interesting and useful properties for humanity. So how do we help the computer to maneuver and sample this chemical diversity? So one thing we tried that Professor Spuruguzic's group has done was to develop a new way of data representation. That is a new type of data structure to represent atoms and molecules. So the conventional one that chemists had been using for a long time is called SMILES. This takes a molecular structure and breaks it down into a simple string. And this string is both relatively human interpretable as well as can be processed by a computer. This is SMILES strings have been around for decades, and that's why people have continued to use it. But Professor Spuruguzic's lab found out that by using SMILES strings, this kind of representation is not very efficient for machine learning algorithms. Instead, they were able to develop a new data structure that they called selfies, which are similar to SMILES, but the main difference is that they have full topological coverage. So it shows the full connectivity of each atom to other atoms in the molecular structure. And what this simple connectivity graph type of data structure does is that it provides more information to the computer, to the algorithm. This is a way of capturing the some of the chemical knowledge that is in a structure. And they were able to show that by using this type of data structure, they were able to achieve with much higher efficiency conventional generative AI methods using SMILES strings. So here, for example, they're looking at a property called chemical beauty, which is a metric for how drug-like a molecule is and how synthetically accessible it would be. And what you can see is that our algorithm starts with a population of molecules that are randomly chosen. So they have very low scores of chemical beauty, and that over many epochs or generations of calculations that they can move the average chemical beauty metric for the population very high. Oh, sorry, it's the green, which is the mean value. So they can significantly improve both the minimum value and the mean of the population. So that was a initial proof of concept. So now the more recent work by their group with our group is using this in a discrete way to generate drug-like molecules. And for this, we are applying genetic algorithms with deep learning. So a genetic algorithm, it's a kind of optimization algorithm. So some of you are familiar with different kinds of optimization algorithms like steepest descents or gradient descent. So these are ways to identify a optima in a functional landscape. So that's quite simple. A genetic algorithms in particular are nice because they can they exchange information between different molecules and to help reach a uh, a new help create new molecules that combine the best features of the prior molecules. And the deep learning comes in where there is a neural network that is trained to distinguish molecules that are good versus molecules that are bad. So you need a training set of molecules that are good to initially train the discriminator, but also the discriminator also learns from the genetic algorithm. So the genetic algorithm comes up with some kind of scoring function, and the discriminator over time uses this along with this initial training to be able to distinguish good and bad molecules. And so they were able to show that using this kind of method for chemistry was able to show rapid improvement in many metrics for molecules, much faster than other methods. And so this is what we're using now in our work with SARS-CoV-2. So we're applying this to both the M-Pro protease as well as the NSP16 enzyme. So here is something that I thought was particularly impressive. They started with just a methyl group in the active site of the enzyme. And over time, the algorithm expanded this methyl group into more increasingly complex and drug-like molecules. So you can see that after 10 generations, you now have relatively small, simple molecules. And then by the time you reach your 25th generation, you have molecules that are similar in size and complexity to drug molecules. So this is ongoing research that we're still evaluating, but what we see now is that it creates molecules that relatively follow the rules of chemistry. But if you are a expert chemist and you look at these molecules, they look rather strange to you. And if you think about them, you can see that these molecules are not molecules that are either unstable or cannot be readily synthesized. So that's a problem. So what we're thinking this is the most useful, at least at this point, is that this can generate ideas for new molecules. And the raw output, at least all at this current time, are not directly useful as drug molecules. But you can use these ideas, and a human expert could use this and say, this part looks kind of bad, but I like this part. And I can simplify this and make it into a real drug-like molecule quite easily. So for example, one can take one of the outputs of the generative AI and look for apply some relatively simple transformations to simplify that molecule. And here in this case, we are able to find an analog of one of the AI molecules in the PubChem database. So this is the largest database of known molecules. It's freely available for you to search. And we see that, hey, this molecule is similar to the output of the AI. And when we perform a docking with this molecule, we can see that it fits quite well, although not perfectly, into the active site of Mpro protease. So this is a good start, and we can use conventional as well as AI techniques to improve and expand this molecule to better fit this binding site, as well as to fulfill other important chemical feature properties such as solubility and non-toxicity. So we're still working on trying to increase the drug likeness of the output from our AI methods, as well as to find ways so that the molecules that it outputs are what we call synthetically accessible. For example, some of the molecules that the AI generated, one of our chemists looked at it and they said, this is an interesting molecule, but it might take me a year to make this molecule. It's too complex. So that's not very useful in a real world setting. So we need to be able to incorporate some kind of synthetic chemistry knowledge into this process. Another thing we're using is incorporating principles from what's known as fragment-based drug design into this AI method. So fragments are basically small, you can think of them as small pieces of drug-like molecules. And instead of just adding on atoms one at a time, we can add on and transform chemical groups in the molecules. So this is a little bit more resembles how real life chemists think and operate. And an interesting idea is whether this can be used as a test of drugability. So for example, our group and other groups, we've been looking at many of the different potential drug targets in the SARS-CoV-2 virus. And it's not clear for especially some of the less well understood proteins, whether they would make good drug targets or not. So if we had crystal structures or good models of those proteins, maybe we can use an algorithm like this to say that, hey, it turns out that this protein, it's not easy to make a good small molecule inhibitor against it. But for this other one, it's quite easy. So maybe that means that would make that other protein a better one for researchers to focus on. Again, we are all pressed on both time and resources, so it's important that we can narrow down our research as fast as possible. And in our preliminary results with these genetic algorithms, with these machine learning algorithms, as well as conventional computational methods, they both point to Mpro protease and NSP16 as being very favorable candidates for drug design. So we are left with some big questions. So some of you are familiar with this kind of artwork where an AI algorithm is trained on human artwork and then is asked to come up with its own ideas. Can we do the same? Can we ask an AI to be trained on chemistry data and to come up with new molecules, especially ones that would not be identified easily by human experts? So this is a kind of computer creativity. Very exciting. So I want to thank the people in our computational lab, a team of postdocs and students from my lab. The experimental work has been led by Dr. Messick at the Wistar Institute in Philadelphia, United States. And of course, I mentioned the important role of the work of Professor Alan Spuruguzik at the University of Toronto, who is the lead on developing computational methods, lead on developing new AI methods for chemistry discovery. And our sponsors, the earliest work was funded by the United States National Science Foundation. Currently, we are funded by Intel, the chip maker. And of course, like I said earlier, we would really appreciate if anyone out there has partners that can help sponsor our research. We're at the point where we think we know how to do the science quite well for this, and we are really just we need more money and we need more manpower to make faster progress. Thank you very much for your time. I'll be happy to take questions.
Sir, thank you so much, sir. Thank you so much. That's an eye-opening session that you have linked the artificial intelligence with drug discovery. And you have given us the deep insights on calculating this protein-drug interactions, protein ligand docking, generative artificial intelligence to create new molecules. And so thank you so much for your time. It's your past bedtime. We know we have troubled you a little bit. And we look forward to conduct more sessions with you. And I'll let you know, just please stay online for a second. I'll let you know if there are any questions from participants.
Of course.
Yes, there is one question. No. Sir, there is a question from Bharti. If someone would like to be a part of the work in your group, how to apply?
Oh, email me and and you can send me a CV or a brief description of what is your background and where you would like to contribute.
Okay. Thank you so much, sir. Thanks for your answer. And personally, I want to ask you a question. Sir, I'm Rohini. I'm CSR SRF from Vignan University. I'm pursuing my PhD here right now, sir. Sir, I'm a little bit interested in your presentation. So, what is the chances of getting postdoc? Like, how to apply postdoc position at your place? And I want to collaborate with you, especially. So, how can I step forward for this?
Currently, we do have a postdoc. I should have mentioned this. Thank you for reminding me. We do have a position open for this kind of work. It's based out at the University of Toronto.
Okay.
And if you go to Professor Aspuru Guzik's website, you will find a link to the application for this position. But this would be a position at Toronto supervised by both Professor Aspuru Guzik and myself and other scientists at Toronto. So this would be a great opportunity for those of you who are seriously interested and have some background in machine learning and chemistry.
Okay. Thank you so much. I'll just check it out. And there is one more question from Vedanidhi. How much of the time is being saved after the introduction of computerized screening in drug discovery and development?
This is an interesting question. This is a challenge for a lot of the field of computational chemistry as a whole, that traditionally a lot of the data and in fact the code for a lot of the software for the research is not shared openly. So I and many others are pushing for open sharing of data and code. So our group, Professor Spurguzik's group, my group, we try to make our code and our data available as much as possible. But there are some problems. A lot of the most useful and widely used software in the field are proprietary and are produced by companies, so they are not so keen on sharing their code. And a lot of the largest and best data sets are produced by drug companies, and they of course do not share their data.
Yeah, thank you, sir. That's a nice explanation. There is another question from Dr. Paul. Can we search for single ligand targeting multiple proteins through AI?
Sure. I don't know of anyone who's working on that, but I don't see any reason why you could not. That's one of the wonderful things about AI is that it is relatively agnostic to what kind of input goes into the algorithm. Anything that can be quantified and you could quantified can be fed into a machine learning type of algorithm, and it can automatically calculate the weights of the different inputs. And so basically, the more information you feed the AI algorithms, at least with high quality data, in principle, the more effective the algorithms should be. And if you feed it relatively, let's say you feed it information that turns out not to be such high quality, in principle, it should have a smaller deleterious effect, as the algorithm should be able to downweight the input of the weighting of the low quality input.
Yes, thank you, sir. There is one more question from Guha. Is there any contribution of nanomaterials in the field of computational biology for the drug designing?
This is a little bit outside of my area of research, so I don't know the details. I know with nanomaterials, most of the focus is in the area of drug delivery, so and drug formulation. So meaning that someone like myself might be able to come up with an active drug molecule, but then in terms of how do we get that drug molecule into a way that is most useful for treating a certain kind of disease, perhaps you might attach that molecule to some kind of nanomaterial, and you can target this nanomaterial for superior to specific types of cells or specific types of receptors, or to increase bioavailability of the drug molecule. This is a very active area of research, but I am not so knowledgeable about it.
Yeah, there is one more question from Swati. Sir, what are the software you have used for the library preparation? What are different ways to understand quantum chemistry using AI? How much time it is supposed to take to complete the drug?
Oh, okay, that's a lot of questions. In terms of the different software packages, Professor Aspur Guzik's group and my group, we tend to develop a lot of our own packages. For docking and virtual screening, we use Smina a lot, SMINA. This is an academic software that is freely available. You can download it from Professor Cole's group. And this is similar to, basically a modernized version of Autodoc Vina that many of you may be familiar with. I also use some commercial software. I like to use the Schrodinger software suite. I know that software is very expensive, and I know a lot of people don't have access to it. That's a problem. But we do try to use freely available software as much as we can so that other people can access the data and share code.
Sir, there are a few more questions. So I want to ask you, like, if you want to end this session with this question, then it is fine. Or if you want to continue, I'll ask the other questions too. So it's up to you, sir, because it's your time.
Yeah, maybe we can take 2 more questions then.
Okay, sure. There is a question from Marlene. Can you suggest how to extract genomic data for working on cancer genomics?
That's also a little bit outside of my area because I don't work in genomics. I work in drug design. But I think one of the most useful areas is by looking at looking at RNA expression levels and correlating that with known drug activity or drug resistance, and seeing or known mechanisms, and using that to identify molecules that act by certain correlating molecules or parts of molecules with different kinds of patterns in RNA expression. So this is this is an active area of research. I don't think a lot of people work on it because it's hard to get access to that kind of data and to have the expertise to work in both fields. But I know groups, for example, at the Broad Institute are particularly active in this area.
Sir, this is the final question of this session. How efficient the supervised learning algorithms like linear regression support vector meshing when it comes to drug discovery?
Oh, I mean, so clearly those methods, a linear regression, of course, that is what we would consider a basic mathematical tool. So that is always used everywhere. You definitely want to be able to compare any kind of machine learning approach you take to something simple like a linear regression model and show that your method can uh, outperform by far a linear regression model. So it's useful as a control experiment. SVMs are still useful in many ways. In general, deep learning has shown, at least if you have enough data, it has been shown to outperform uh, SVMs and other methods like random forest methods in almost all areas. But in a case where you don't have so much data, SVMs and random forests can outperform neural networks.
Sir, once again, we wholeheartedly thank you so much for your time and patience, sir. We look forward to conduct more sessions with you. Thank you so much, sir. Thanks again.
Thank you very much for participating. I look forward to hearing from anyone who has further questions or interest in this field. Thank you very much.
Okay, participants, now we'll take a break for 10 minutes and then join again.
Hello, sir. Hello.
Yes, sir. 2 minutes. 2 minutes, sir. Shahul, sir.
Yeah. Hello, sir. Yeah, please. Hello. Yeah.
Yes, sir. 2 minutes. 2 minutes.
Yeah. Hello. Yeah, please. Hello. Yeah.
2 minutes.
Participants, welcome back after a short break. And now I'm here to introduce the next speaker, Dr. N.S. Shahul Hamid. Dr. N.S. Shahul, scientist, Vice Cornell Laboratories Private Limited. Dr. Shahul did his PhD degree from the discipline Structural and Computational Biology in the year 2011 from Indian Institute of Science, Bangalore, and MTech in Biotechnology from Center for Biotechnology, Anna University, and B Pharmacy Pharmacy from Dr. MGR Medical University. His work experience include Associate Professor B.S. Abdur Rahman Crescent University, tutored Gate Forum, Coimbatore, Assistant Professor Bannari Amman Institute of Technology, Postdoctoral Fellow Tata Institute of Fundamental Research, Postdoctoral Fellow Indian Institute of Science, and Research Assistant Indian Institute of Science. Now he is a CEO in Vice Cornell Laboratories, Chennai. He has also given coaching on CSIR, UGC NET, and GATE Biotechnology Life Science to many university students for the past 4 years. His area of expertise include bioinformatics and computational biology, proteomics and genomics, fermentation technology, bioprocess technology, structural biology, molecular biology, pharmaceutical biotechnology, genetic engineering, and gene manipulations. He has 5 publications and has participated in 2 workshops, in which one workshop he is served as resource person and guest lecturer for international workshop molecular modeling and simulations in drug design and protein engineering held at University of Nottingham, United Kingdom in the year May 2017, and 12 other workshops at national level. And his awards and honors include he has a GATE score of 98.72 percentile, that is All India 41 rank, qualified Indian Council of Medical Research JRF, qualified NET, qualified Combined Biotechnology Entrance Exam for admission into MTech program in Chennai, and have received Erasmus Fund for academic visit to University of Nottingham, United Kingdom, 2017. Apart from all of this, he's an energetic and positive person, and he is there with us today to give a talk on our workshop. Now, without further ado, I would like to welcome Dr. Shahul. Sir, please give your talk.
Yeah, please. Thanks for the very elaborate introduction. So I thank the opportunity given to me by Vignan University. I thank Professor and HOD Kripanadi, Assistant Professor Abraham, and the chairman and other management of Vignan University for providing this opportunity to be the resource person for this workshop. So with this I start. I'll share the screen. Okay, just 1 second. Is my voice audible? Are you able to hear me? Hello?
Yes, sir. Yes, your voice is audible.
Okay. Yeah, so thanks for the introduction. I like to thank the Vignan University for giving this opportunity. I like to thank Professor Kripanadi, Professor Venkateshwarlu, and Professor Abraham for giving me this opportunity to conduct this workshop as a resource person. And I also thank the management of Vignan University to, give me this opportunity. So I would like to talk on AI-powered drug discovery. So can I share my screen, madam?
Yes,
Yeah.
sir, please.
Yeah. So yeah. So are you able to see my screen?
Yeah, yes,
Hello?
sir. It
Yeah.
is. Yes, yes.
So I'm Dr. Shahul from Vice Cornell Laboratories, and I'm going to give a workshop and technical seminars on AI-powered drug discovery and development. Okay, particularly COVID-19. So the thing is, we are going to talk about how artificial intelligence tools are used to accelerate the drug discovery program against this COVID-19 pandemic. Okay, so here, like this COVID-19 has created a situation where, like people are, particularly the pharma companies are trying to, work day and night to come up with a drug to prevent the disease from actually causing this havoc. So people are trying from various fronts. People are trying to develop vaccines. People are trying to develop drugs, small molecule drugs, okay, against various COVID-19 targets. And people are also trying to come up with, other omics data related to this COVID-19. For example, the genome sequence of COVID-19, the susceptibility of patients to COVID-19, their genomic sequence, and all those things. So here, a multi-faceted approach to manage the situation of COVID-19 is going on. Okay, so here now the theme of discussion here is artificial intelligence powered drug discovery and development against COVID-19. So first we have to understand what is artificial intelligence, what is machine learning, and how they are used in drug discovery programs. What stage they're used, where they're used, and what will be the outcomes. So this is what this technical seminar series over these 5 days we will be discussing. So right from the initial stage of drug discovery till the end, artificial intelligence tools can be applied for accelerating the drug discovery program. Okay. So now here, when you talk about artificial intelligence, so before going to the topic, I will just like to define certain terms to make things easy. Okay. So what is artificial intelligence? It refers to the simulation of human intelligence in machines that are programmed to think like humans and mimic their actions. So here you have the machines which can be made to think like humans, which can be programmed to think like humans, and it can mimic human activities. So this is what is the broader sense of artificial intelligence. So it is actually can be defined as a program that can sense, it can reason, and act and adapt. Okay, so this is what is actually called artificial intelligence. Now there are various tools used in artificial intelligence, various techniques are there. For example, the most widely used is the machine learning approaches. So machine learning approaches means is the application of AI. Machine learning is an application of AI that provides systems the ability to automatically learn and improve from experience without being explicitly programmed. So here machine learning techniques, they learn from the data, they learn over time when they're exposed to data, and these algorithms get refined more and more as they're exposed to more and more data over time. So here they can learn from experience, so without being explicitly programmed. Okay, so that is the definition of machine learning. Okay, so various machine learning techniques are used, like neural networks, support vector machines, random forest, linear regression models. Like we can go on. The several techniques are used for machine learning. So out of which techniques like deep learning are making a lot of news now. Okay, so it's a subset of machine learning. Deep learning is a subset of machine learning in which multi-layered neural networks. Okay, so multi-layered means several neural networks are combined. So multi-layered neural networks learn from vast amounts of data. So here, for example, you would have heard about examples where Facebook uses facial recognition algorithms. Many companies, they use facial recognition algorithms. So they are based on neural networks, deep neural networks. For example, convoluted neural networks. So these neural networks are able to gather data. They are able to finally give output in terms of identifying, recognizing patterns, and then they are able to even recognize face patterns and recognize the person. So there are different applications of deep learning. Now these deep learning approaches are now being used in drug discovery programs. Okay, for example, predicting the structure of target proteins. Okay, so that is where deep learning is now used in drug discovery programs. Now, if you see the last few years, maybe the last 1 or 2 years, there has been actually a really explosion of startups. Several startups have come up. Nearly some 200 to 300 startups have emerged, which are using these artificial intelligence and machine learning, deep learning, these kind of techniques. Okay, these kind of strategies to accelerate drug discovery program. Okay, so you must have heard about really companies like DeepMind, companies like Atomwise. They are actually like companies which are actually based on AI, and machine learning, artificial intelligence, machine learning, and they are helping to accelerate drug discovery program. Okay, so several startups, even in India, are there, which actually are trying to help in drug discovery, accelerate drug discovery by using these artificial intelligence tools. Okay, so now we are at a stage where we have defined what is artificial intelligence, what is machine learning, what is deep learning. Okay, so as we, through the different talks, as we will, like tomorrow, day after tomorrow, as we go through, I think the meaning of these will be understood as we go. Okay, so now what we can do is just look at the drug discovery and development timeline. Okay, so now typically, if a drug has to be approved for human use, a novel drug has to be approved for human use. Okay, for example, imagine like that a chemist is trying to discover a scientist is trying to discover a drug against COVID-19. Okay, so it will take several years down the lane in order for that particular drug to reach the market and to reach the end user, that is the patients. Okay, it will take several years, maybe like 12 to 13 years it may take, or even more. Okay, so the reason is several drug discovery process, several compounds have to be screened. Okay, so screening takes a lot of time, and then they have to be tested against for example, enzyme assays, cell-based assays, all these kind of preclinical studies have to be done, and then toxicity and safety has to be evaluated, and then like they have tested in animal models. Okay, so again, like in animal models, toxicity and safety has to be evaluated, efficacy has to be evaluated, and then once it actually, gets approved for clinical use, clinical trials. So once the preclinical data is promising, okay, so those lead compounds will be taken for clinical trials. Okay, So in clinical trials, generally few compounds only actually qualify to be taken for clinical trial. Okay, and it's very expensive process, clinical trials, and also time taking process. It may take several years, 7 years or 8 years for clinical trials to get completed. So it needs human volunteers. Okay, so here the clinical trials itself is actually like phase 1 trial, phase 2 trial, phase 3 clinical trial. Like that, it's actually divided. So phase 1 trial generally employs healthy volunteers, and here the drug is given to a small set of healthy volunteers, where the only purpose is to test the safety of the drug. Suppose healthy volunteers, they develop some symptoms, some acute poisoning or some toxicity because of the drug intake, then they are actually the drug is stopped there. So suppose the drug actually shows no toxicity on those volunteers, then it's taken to the phase 2 clinical trial. So in phase 2 clinical trial, patients who are suffering from disease, okay, they are actually given the drug. So there, what happens? Different age groups differ. For example, people of male gender, female gender, different age groups, and different body weight. So they are taken group, and then the trials are conducted there. Okay. So so the thing is, once the phase 2 trials, in phase 2 trials, the efficacy of the drug is evaluated. So then actually in phase 3, they will try which formulation will be better, which dose is the correct one to give. All those things actually are studied in phase 3. Okay. So finally, in phase 3 is conducted in a large group of patients. Okay. So and generally the toxicity and long-term chronic toxicity is evaluated. At the same time, the efficacy and how the existing treatment, okay, how the new drug actually has advantages of existing treatment, that is also evaluated. So finally, these clinical trial data is submitted to FDA, okay, Food and Drug Administration. So the FDA will actually take nearly 1 to 2 years to go through those clinical trials and check it, okay. And after elaborate checking, what they will do, they will suppose that they are satisfied with the clinical trials, and then they will grant FDA approval. So all these things will take several years down the lane, some 12 to 15 years it will take. So now we are actually facing a situation where COVID-19, this SARS coronavirus version 2, is spreading like rapid fire, and it is going through spreading throughout the world, and it's causing a lot of disease, lot of havoc. Now people are dying because of it at alarming rate. So since the infectivity of this particular version of SARS is quite infectious, so it has become a pandemic. That means it is spread throughout the world. Okay, like very few countries are exempt from this virus infection. So due to this reason, there is the urgent need. There is a very urgent need to come up with drugs which can actually counter this disease, counter the virus. So now the typical routine of drug discovery and development timeline cannot be followed. So people cannot wait for 12 years. So the thing is now people have to be smart, and then they have to employ artificial intelligence tools in order to accelerate the drug discovery process. That means we have to shorten this, like from 12 years, you have to shorten this to a few months to develop the drug. Okay. So that is where AI can come in. So this is the opportunity for artificial intelligence tools to come in, so that the drug discovery process is actually completed in a shorter duration of time. Now, where these artificial intelligence tools can be applied? So wherein can be applied? So one is actually right from the start. Okay. For example, you have this COVID-19 virus. Okay, the SARS-CoV-2 virus. If you take the SARS-CoV-2 virus, it contains several. It codes for several proteins. Okay, some are structural proteins, some are non-structural proteins, and now the thing is identifying those proteins which can be suitable targets for drug design and development. Okay, so inhibiting those targets should end in the viral being inhibited, virus being inhibited. So the thing is now people want to, find the structure of these proteins, viral proteins. Okay, they want to once they are actually armed with the structure of the viral proteins, they can actually design drugs. Okay, so because now structural biology based drug design, rational drug design, okay, is a promising field. So in order to rationally design a drug, we need to know the structure of the protein. Okay, at atomic resolution, three-dimensional structure of the protein at atomic resolution is needed to design drugs. So thing is, like bioinformatics already played a role in accelerating drug discovery. By using computers, people are now able to design better and better drugs with less toxicity, okay, with better pharmacokinetic profile than before. Okay, so instead of randomly searching for drugs through computer library and all those things, now people are able to gear in and save time by using a rational approach of structural biology based drug design, structure based drug design, where the protein structure needs to be known. Okay, so here traditional methods like X-ray crystallography and NMR, okay, those are the methods by which the structure of the protein, the target proteins, okay, can be elucidated. For example, suppose out of the many proteins which the COVID-19 makes, there are proteins like this RDRP, RNA-dependent RNA polymerase, okay, which could be a crucial target. Similarly, the protease, the main protease, otherwise called as the 3-chymotrypsin-like protease, that also has been identified to be a major target for drug discovery. Similarly, papain-like proteases and other proteins, okay, within the COVID-19 genome, they have been now, like for example, the spike glycoprotein, okay, that also will be termed as one of the important targets against which drug design can be started. So identifying these targets and then elucidating the structure of these targets is very important for designing drugs. Okay, so with this in mind, we have to really come up with a way to get the structure of these proteins in a very shorter duration of time. So anyway, advances in existing structural biology methods like X-ray crystallography and NMR, okay, have paved way for quick, almost very quick, structure elucidation of these proteins. Okay, for example, years before, when Max Perutz and John Kendrew, they started the structural elucidation of hemoglobin. Okay, it took 21 years for them to solve the structure of hemoglobin. Okay, so that was one of the first proteins whose structure was elucidated using crystallography. It took 21 years for them, and mainly because that time recombinant protein expression was not there. So, and then the protein purification strategies were not there. Computational, graphics was not there. Computer graphics was not well developed at the time. And then all these softwares, okay, which are used in crystallographic refinement were not there. Okay, so it took several years, like 20 years down the lane, to get the structure of hemoglobin. Now, recently, people have solved the structure of the SARS glycoprotein. Okay, that SARS glycoprotein, the spike protein receptor binding domain in complex with the human AC2 receptor. Okay, so that structure is available in PDB, and they were able to get it within a few months. Okay, that means now people are able to get the structure of protein, the two protein complexes, in a matter of few months. This is because of improvements in the recombinant expression of proteins, purification strategies, improvement in purification strategies, enabling them to get more proteins for crystallography, and then better algorithms and computer graphics, which help in really accelerating the structure elucidation. Okay, so the thing is now people are able to get the structure of these proteins in very short time. Okay, so now this COVID-19 situation has put has made the COVID-19 pandemic has created a situation where even this is not enough. Okay, that means even the crystallography, even though now it is quite quick and is able to get the structure of the protein in a few months, still this is not enough. That means now, if I want to know the structure of all the proteins in the COVID-19 genome, okay, coded by the COVID-19 genome, then it may take several years down the lane. So, in order to accelerate this, people are going for prediction methods. Okay, prediction methods, but to predict the structure from the sequence of the protein sequence, you need homology modeling, these kind of tools. But these kind of homology modeling and other techniques, they are based on similarity, at least 30% identity between the template structure and the query protein. Okay, sometimes you may not have even 30% identity. So these kind of novel sequences, it's very difficult to find the fold of the protein. Okay, so now in order to solve these thing, people have come up with AI-based tools like AlphaFold. Okay, so AlphaFold is an AI-based tool which has come up recently. I think this January 15th only the paper got published in Nature. So the AlphaFold is a deep learning and neural network based algorithm in which actually tries to predict the structure of the protein directly from the sequence without using any modeling strategies. Okay, so here a group of machine learning experts working in DeepMind, the company called DeepMind, they were able to come up with an algorithm called AlphaFold, which is able to predict the structure of the proteins with very reasonable accuracy. Okay, so in fact, last year, before the paper got published, last year somewhere in November, there was actually a competition called the CASP13, Critical Assessment of Protein Structure Prediction. 13th competition was held. So in these competitions, big giant companies like from Pfizer and Novartis and all these multinational companies, they came up with some algorithms to predict the structure of proteins directly from sequence. Okay, but actually they failed to actually qualify in that CASP13 assessment. Whereas this newcomer, DeepMind's AlphaFold, which
Sir,
is
are you in which slide, sir? Shahul, sir?
yes, sir.
I can see the drug list.
You want to show slides? Okay. So
Okay, no problem.
Yeah.
Okay, continue, sir. Sorry.
Yeah, I'll just show this. So this AlphaFold, yeah. So here AlphaFold, this is actually a machine learning artificial intelligence based tool in which is able to predict the structure of proteins with very high accuracy. Okay, so this has really come up, to use in the latest situation where AI has been used to predict the structure of these proteins. In fact, you can see that this AlphaFold has already predicted structure of several COVID-19 targets. Okay, so we can just type AlphaFold COVID-19 structures. You can see several blocks. So you can see here in DeepMind, this is a company which actually has written this algorithm called AlphaFold. So you can see computational predictions of protein structures associated with COVID-19. So here, in order to accelerate the drug discovery program, the first step that one can take is getting the structure of the target proteins known, okay, at least predicted with high accuracy. Once these target structures are, validated, okay, then drug discovery can start. People can do virtual screening. People can try to dock ligands from the library against these targets. Already FDA-approved drugs can be, which can be repurposed against COVID-19, can be tested on these targets computationally. Okay, so they can dock all these FDA-approved drugs on these targets. Okay, in order to come up with a, possible cure for this COVID-19. So here, as it takes several years down the lane for FDA to grant approval to a novel drug, people are thinking about repurposing existing drugs. So already drugs which are in the market, which have been evaluated for safety and which have been, considered to be safe and least toxic to humans and which have got already FDA approval for human use, these drugs are now repurposed for COVID-19 situation. For example, HIV protease inhibitors. You must have heard about the HIV protease inhibitors. These were one of the first few rationally designed drugs. Okay, the protease inhibitors of HIV were one of the rationally designed drugs. Okay, so nelfinavir and lopinavir, this kind of drugs actually which were designed originally to be used against the protease of HIV has now been found use against COVID-19. Okay, so COVID-19 protease also can be inhibited by these drugs. So they've done molecular docking and they've proved this. So here, getting the structure of the main protease, okay, so that is a very, very important. And luckily now the structure of main protease is available. Okay, you can go to PDB and find out. Already in Protein Data Bank, you have a COVID-19 resource where at least 3 important target proteins have been structurally determined through experimental methods. Okay, that is, one is the spike glycoproteins receptor binding domain in complex with the AC2, angiotensin converting enzyme 2. That structure is available in PDB. So this is solved by crystallography. The other thing is the main protease, the chymotrypsin-like main protease of COVID-19, which is one of the important targets. Okay, that is also available in PDB. That is also determined by crystallography as well as by cryo-electron microscopy. Now, recently, people have determined the structure of the non-structural protein 12, NSP12. Okay, NSP12 in complex with NSP7 and 8. Okay, and in complex with a drug called remdesivir. Okay, is available in PDB now. So I will just show the PDB. Go to PDB. So PDB is Protein Data Bank, and you can see this RC is a PDB, Royal Chemical Society of Britain PDB. So if you just type NSP12 of COVID-19. So if you can see NSP12 means, yeah. Can you see here 2020? It got published in Science. SARS-CoV-2 RNA-dependent RNA polymerase in complex with cofactors. So you can see that various structures are there. Okay. So here using electron microscopy is determined. Similarly, you can see in complex with remdesivir. NSP12 COVID-19 remdesivir. Okay, can you see here the non-structural protein 12, non-structural protein 7, and non-structural protein 8 complex bound to the template primer RNA and triosphosphate form of remdesivir. So this is actually a drug. Remdesivir is a drug which inhibits the RNA-dependent RNA polymerase. Okay, which is a very crucial enzyme for this particular COVID-19. Okay, so this particular remdesivir is also a repurposed drug. It has already been known to inhibit the SARS-2003 RNA-dependent RNA polymerase. Okay, so this drug is now when they tested against COVID-19, they were able to actually get some promising results. Okay, so still it's ongoing. Still the research is ongoing. People are trying and testing these drugs against various clinical trials are going on. Okay, but these are all already FDA approved drugs. Okay, so already safety is actually guaranteed. But one thing is whether they will turn out to be highly efficacious or not. That is what we have to see. Okay, so you can see here this 7BV2 structure of NSP12. NSP12 is nothing but non-structural protein 12. That is nothing but the RNA-dependent RNA polymerase. This is the enzyme RNA-dependent RNA polymerase requires other proteins, other viral proteins to do its work. That is the replication and translation machinery. So here NSP7 and NSP8 are actually smaller proteins which work in conjunction with NSP12 to achieve this task. Okay, So this is inhibited by the drug called remdesivir. The structure is available. Okay. So you can see the cryo electron microscopy is available, and you can see the main protease of SARS-CoV-2, main protease of SARS-CoV-2. You go to main protease of SARS-CoV-2. That structure also is available in complex with various inhibitors. You can see the crystal structure of COVID-19 main protease in complex with carbonophore. That is a type of drug. Okay. So structure of SARS-CoV-2 main protease in complex with UAW243. UAW243 is a peptidomimetic drug. It's a peptidomimetic means what? Is basically a peptide which binds to the active site of the protease, and it's a non-cleavable peptide. It's a peptidomimetic. Okay. So here UAW243 is a small peptide. Okay, basically a peptidomimetic. Okay. So here such as now determined by crystallography, X-ray diffraction at a very high resolution, 1.7 Armstrong resolution. Okay. So now you can see in 2020 so much, just in a matter of few months, the SARS COVID-19 itself was actually known to scientists only in late November or December, basically December 2019 only it was known. Okay. Within a matter of few months, okay, they were able to get the structure of these target proteins. This is because of the improvements in structural biology and bioinformatics. Okay. Now, even this is not enough, because right now people have got the structure of only very few proteins of COVID-19. What about the remaining 20 or 30 proteins still whose structure is not known? Okay, there are many other proteins in COVID-19 whose structure is still not known. Okay, so in order to accelerate this process, we need to actually use computational tools, particularly artificial intelligence based tools, to accurately predict structures. Okay, so that is why this AlphaFold has come in. So if you take AlphaFold, this AlphaFold actually tries to solve the protein folding problem. In that, it is able to get the 3D structure of the protein predicted directly from the sequence of the protein. Okay, so this, can you see here, the amino acids fold into helices and other sheets, forming secondary structures, and secondary structures themselves, they fold to optimize and lower their energy, giving rise to tertiary structures. And tertiary structures may, in fact, actually, after getting folded, they may interact with one another through protein-protein interactions to form quaternary structures. Okay, this is how the protein folding goes on. So here, directly from the amino acid sequence, we are going to the final stage. Okay, through AI-based prediction. Okay, so what is the method used? Okay, so if we want to know what does this deep mind, what is the algorithm this deep mind has developed? Okay, in order to make this prediction, protein structure prediction, that is nothing but a deep learning neural network. Basically, a deep learning convoluted neural network. Okay, this convoluted neural network was the same convoluted kind of neural network which has been applied in facial recognition. Okay, pattern recognition, facial recognition is already in use. Okay, many companies are already using them for facial recognition, like Facebook and other companies. Okay, now this has been modified, and deep learning concepts have been applied. The deep convoluted neural network has been developed in order to now predict structures of proteins. Okay, so here we can see that one of the target proteins whose structure is got directly by folding the protein from the sequence. Okay, so this particular algorithm is able to fold the protein directly from its primary structure, that is a sequence, into a final fold it's able to predict. So this is actually an animation of how the AlphaFold works. Okay, so if you see machine learning and AI has been in use years before. Okay, it's not a new thing. It has been in use years before, but now it has become more and more sophisticated. If you know, machine learning is already there in your emails. Okay, how the spam checking is done, how all this kind of spurious and unwanted emails, how they get automatically filtered and go to spam. It is because of machine learning. Machine learning algorithms have been used in order to achieve this. Okay, so spam detection and filtering of unwanted emails is an application of machine learning. This is a trivial example of a machine learning. Advanced examples include these kind of things. Okay, so the machine learning itself has developed over years that it now is able to come up with structure of proteins directly from the sequence. Already we have heard about self-driving cars. Okay, Uber and other companies are now launching self-driving cars. Again, the self-driving cars are based on very sophisticated artificial intelligence tools. Okay, but you have heard about even the COVID-19 alert systems, where tons and tons of literature are scanned for COVID-19 reporting, and then it is actually the entire world is alarmed. Okay, but through these AI-based companies. Okay, for example, there's a company called Blue Dot, which is scanning very voluminous data published across various languages in various countries related to COVID-19, and then it's actually broadcasting to the public. Okay, so and making them alert not to visit those countries. So this is actually or different applications of AI in various fields. Now, time has come where artificial intelligence has now been applied to structural biology. Particularly, they are now helpful in elucidating the target protein structures, and now you are actually able to fold the protein to three-dimensional folding of the protein is done directly from the sequence, and that uses something called deep convoluted neural network. That is the algorithm used. Okay. So apart from that, there are many other neural networks, many other artificial tools which have been now used in various aspects of COVID-19. For example, based on the different symptoms known, okay, now AI-based programs can tell whether a patient has COVID-19 or not. So these are based on classifiers. Certain classifiers are used to classify the patient as COVID-19 or not from the clinical data and from the symptoms exhibited. So they are actually having higher accuracy of prediction than just by using medical data. Okay, and in fact, like for example, in medical diagnosis, already artificial intelligence has made a very big impact. Already people are now well aware how cancer detection and other disease detection can be done at a very early stage using AI-based tools. It's already there. Okay. Now people have used similar medical diagnosis against COVID-19 also, like from the patient symptoms exhibited in patients to the clinical testings to there are certain algorithms which can just tell whether a patient is infected or not from the facial patterns. Okay, so various techniques have been developed, and they are coming to meet the COVID-19 pandemic. They are able to now address this situation. If you see, like many robots have been employed. Many robots have been employed in airports and other, clinical settings, hospitals, where the robots themselves act as nurses and go and deliver drugs to the patients. Okay, this is also an application of artificial intelligence. But anyway, our discussion will be in the future 4 days. Next 4 days, it will be basically how AI is used in drug discovery, particularly how the target structure is determined, how the virtual screening using AI-based tools, deep screening techniques are used, how one can actually predict the molecular properties of drugs. For example, suppose from the structural scaffold, whether one can say whether it can inhibit this particular protein or not. So we can do molecular property prediction, toxicity prediction, okay, based just on the structure of the drug. We are able to now say whether the drug will actually bind to the target, whether it will inhibit it, whether it will show toxicity, whether it will show adverse effects. All these can be predicted much in advance, okay, using AI tools. What you need is the structure of the target. Well, structure of the target, structure of the protein, structure of the drug. So once you know structure of the drug, you'll be able to get a wealth of information. Okay, you'll be able to say whether it's going to bind to the target, whether it's going to show adverse reactions. All these can be now predicted with some reasonable accuracy. Okay, so now it's getting better and better as more and more data is exposed to these AI tools. It is getting refined and it's learning and it's improvising itself, and then it's making the prediction better and better. Okay, so now I'll just play on very simple, video which is already available in the internet about AlphaFold. This is actually not a very serious scientific video. It's just a video where a novice gets exposed to AI and how AI is used in structural biology for getting structure of proteins. Okay, so AlphaFold. If you go to, yeah, so I'll play this video.
Spare change, sir. Sure. Wait,
Were you able to see
aren't
this?
you a world-renowned biochemist?
I was, until AlphaFold replaced me.
Hello, world. It's Siraj. In the AI research lab, DeepMind just made the code and data set for their state-of-the-art protein folding system titled AlphaFold, publicly available for anyone to use for their own projects. So I tested it myself on my own laptop, a MacBook with a broken key that I haven't fixed in 6 months. And using it, I was able to predict and visualize different protein shapes given genomic sequences. Now I'm going to show you step by step how to do the same thing on your own computer, from installation to visualization, and of course, explain how it all works. Once you understand it and can run it for yourself, you'll be able to use it for your own applied AI project, be it in health, sustainability, or materials design. And that project will look very impressive on your portfolio because using AlphaFold, DeepMind, a company with 0 credentials in biochemistry, won first place by a large margin in a biannual protein folding competition called CASP, beating 97 other groups, including many billion-dollar pharmaceutical companies who've been working on this for years. AlphaFold, like the ice cream cone rotator, is the definition of disruptive technology. And since it was just made public, you now have this incredibly powerful tool to use for your own projects, or even to compete in the next CASP competition in a few months, since it's open to anyone. For decades, biochemists have been trying to be able to predict how a piece of genetic code will fold into a protein, a very intricate 3-dimensional structure. And this is called the protein folding problem. Most people, myself included, aren't thinking about this problem at all. In fact, for most of my life, I just considered proteins to be a nutrient that helps build muscle. But it turns out that proteins are so much more important than that. In fact, the word protein is derived from the Greek word protos, which means of primary importance. And that's a perfect description because almost every biological process that's required to keep you alive is carried out by proteins. They are nature's own machinery. Think of them as evolution's nanobots. Researchers have estimated that there are between 10,000 to several billion different protein species, and each of them perform a different task in your body, from digesting food to fighting infections. And the reason these proteins are able to accomplish such a wide variety of different, very specific tasks is due to one simple property that they all share: they fold. The process works like this. If we peer into one of the 30 trillion cells in your body, we'll find its instruction set encoded in DNA strands via 4 letters, A, C, T, and G, which stands for Welcome to Gattaca. Different combinations of these letters represent the features of living systems, similar to how 1s and 0s do the same for computer systems. In this genetic code, groups of 3 nucleotides define amino acids, the building blocks of proteins, and there are 64 possible combinations of them. Given a certain amino acid sequence, a protein will begin to fold into its unique shape, a process that aims to target the most stable structure via the path that requires the least amount of energy. We can visualize this process using an energy landscape. As the protein folds, think of it like a ball rolling down a pit of despair. Just kidding. It rolls, it navigates around bumps. These bumps are paths that require extra energy, so it avoids those until it finds a point with the least energy. Does this process sound familiar? It's similar to how neural networks learn the process of gradient descent. They try to find the network values that will minimize not energy, but an error value. And as it turns out, this is the optimization strategy that AlphaFold uses. So the goal is to take as input a genomic sequence and then output its predicted protein structure. This protein structure can be visualized in either 2 dimensions or in 3 dimensions using 2 key features: the distance between amino acid pairs and the angles of the chemical bonds that connect those amino acids. The most successful techniques in this space before AlphaFold involved taking fragments of different protein structures from a protein database and assembling them using an optimization technique called simulated annealing, which unfortunately doesn't involve a ninja sword. This is a process that involves guessing a protein shape, then finding correlations between different protein sequences to iteratively refine its structure. This process works well enough, but can be computationally inefficient because the search space of possible structures is so vast. Another successful technique has been to compute what's called the multiple sequence alignment, or MSA. The idea there has been to find similar protein sequences from a protein database, then align them with the target sequence. Once aligned, they can be used as input to a neural network, which will output predictions for where certain amino acids make contact with each other. AlphaFold used 3 neural networks during the training process. The first neural network, a convolutional network, predicted distance pairs and angles. The second convolutional network estimated the accuracy of a given protein structure. And the third, a variational autoencoder, generated a protein structure. But to be totally frank, it's unclear if that's exactly how it worked. That's just my best estimate after analyzing their documentation. They submitted several papers, one to Nature and one to Proteins, but both had some contradictory information. And the neural architecture described in both papers was also clearly different from the programmatic architecture that they released on GitHub. Hey, where's that fancy variational autoencoder you mentioned?
Growing businesses need a hosting solution that is powerful yet easy to manage. Let's look at how business hosting is a simple yet powerful.
Okay, you saw the video, right? Hello?
Yes, sir.
Yeah, so I think it was just a funny video about how, new technology like AlphaFold is now threatening the, existing structural biology framework. Now, in a few years, it will be possible that, people can predict with very high accuracy the protein structures, and to such an extent that the structural determination methods may become obsolete. Okay, because when you can predict with the same accuracy as determining the structure, okay, then actually people will go for the quicker method, that is actually using AlphaFold to predict the structures, and then they can start the drug discovery program. Now already to the proof of concept, people in DeepMind, they have come up with this protein structures of COVID-19. Okay, so you can go and read here. You can even view the source here. So people already, you can download the structures. Okay, you can open source links. You can go and download the structures. Already several structures of this COVID-19 proteins have been predicted using AlphaFold, and it is there. Okay, so you can download and then you can start your docking or any virtual screening, whatever you want. Okay, so you can read this whatever this here, and you can, as we see here, as we continue to improve our AlphaFold system, we are releasing our most up-to-date predictions of 5 understudied SARS-CoV-2 targets. while people have been concentrating on other targets like NSP12 and main protease, other targets like which are very difficult and almost very not possible in a few years. For example, the membrane proteins. Those membrane proteins of COVID-19, they may be very crucial targets, but expressing the membrane protein and purifying the membrane protein and determining the structure of the membrane protein may take years. Okay, so instead of waiting for that, we can always use this AlphaFold, get the structure predicted with very high accuracy, and then you can go for the other steps in drug discovery. That is virtual screening, docking, getting the binding scores. Once the binding scores are good, you can actually filter them based on the pharmacokinetic profile. So previously people, what they do, they used to make those drugs. Okay, they used to laboriously synthesize. It was actually a synthetic chemist job. So they used to synthesize several combinatorics they use and create a library of, synthesize a library of drugs. Okay, and then they take each of them and test against the target. Okay, mostly cell cultures and all those things. But you know, the cost involved is enormous and time also is enormous. Each drug has to be synthesized and tested against the cell culture. But the thing is, now you can do the same thing computationally. Okay, so you have virtual screening methods, and now these are actually powered using deep learning based AI, which actually improves the accuracy several fold. Okay, several fold improvement in accuracy in screening. Virtual screening is actually achieved. Okay, so over these few days, I will expose to you the various available programs and web servers and tools, AI-based tools, which can help in these drug discovery process. That is, you know, virtual screening, this molecular docking, all powered by AI. Okay, and then molecular property prediction, toxicity prediction. Okay, and then QSAR model, building a QSAR model. Okay, so by using deep neural networks again, QSAR models can be built. Okay, so this can pave way for better drugs to be discovered in a quicker time span. Okay, already you can see the news. For example, if you just type COVID-19 drugs, okay, searched by AI, you can see several drugs are now available for testing, which are actually based on AI predictions. AI prediction COVID-19 drugs. So you can read these papers. Okay, so several papers have come up where AI-based tools have predicted certain drugs. Okay, which these drugs are already in existence, but the AI-based tools have predicted these drugs to actually have better efficacy than the other ones. Okay, so the thing is, people now can actually save time by testing these drugs instead of testing all the available FDA approved drugs. Okay, so that is the thing. Now you can see that several, even if you see, even in Delhi, IIT, they are actually come up with a AI model to repurpose existing drugs. So you can see news. Atomwise, which is one of the leading companies which is using AI against this drug discovery. Okay, so this company called Atomwise leverages AI to explore a broad spectrum of COVID-19 drugs. So here now you have a lot of companies working on AI-based drug discovery against COVID-19, and what we will do in this program is trying to understand how these work. Okay, for example, when you say neural network, how it works. Okay, so what are the types of machine learning algorithms available, and how they're able to predict, how they're able to classify, and how they're able to get a particular value. For example, if we say this particular drug has this much particular affinity. So how am I able to predict that? So these are based on the machine learning algorithms which are used. So there are various types of machine learning algorithms which are used. One is the naive-based classifier, k-means clustering algorithm, support vector machine algorithms, linear regression, logistic regression, artificial neural networks. Extension of this artificial neural networks is the one called deep neural networks. Okay, that is what is used in this AlphaFold and all. And random forests, decision trees, nearest neighbors is some of the few machine learning algorithms which are in current existence. Okay, which actually are widely used now. Okay, for various purposes. Okay, for various purposes. And what we will try to explore is which algorithm is suited for what kind of situation. Okay, so for example, depending on the need, different algorithms are used. Each algorithm has its own advantage. So we'll try to explore in those coming few days, what algorithm is suited for what. Okay, so I think now you got a basic idea about this AI-powered drug discovery. Okay, so still we just not started. And what I want to say is the next few days, the next 4 days we have, in that we will try to explore these in greater detail. Okay, so I will like in tomorrow's session, I will tell about how the deep convoluted neural networks work and how the AlphaFold, okay, is able to predict structures with high accuracy. I will tell those things. Okay, so if you want, you can go to GitHub and download AlphaFold. Similar code is available, not the exact code, but a similar code is available for users to download and practice. But anyway, it needs exposure to machine learning. For example, if you want to learn machine learning, you should be exposed to Python programming. You should know about TensorFlow. You should know about PyTorch. You should also know about the hardware used for AI. For example, the GPU, the graphical processing units, and the other things like Jupyter notebooks. Okay, so these are the tools which are really used in machine learning. Okay, for example, if you want to practice machine learning, you want to code them, you need these tools. Okay, so we will give an exposure to these also. Like what are the tools available? Like for example, Jupyter notebooks, how it can be used, how we can code, how we can. So a basic introduction to these also will be given over the few days that we're going to see. Okay, so with this I will try to stop my lecture. Okay, it's now 12 o'clock. I will try to stop my lecture. So basically today was an introduction to AI in drug discovery. Okay, so from tomorrow we will take each individual case. For example, target structure prediction using AlphaFold, molecular property prediction, toxicity prediction, QSAR model building, even synthetic planning. For example, if you want to synthesize a particular drug, the synthetic planning approach also can be guided by AI. So all these things we will explore in greater detail in the coming sessions from tomorrow. Okay. So with this, I will wind up for today's session, and we will try to meet tomorrow. Okay. Hello, sir.
Yes. Thank you, sir. Thank you so much. That's an eye-opening session. Thanks for your time and patience. Sir, now I would like to ask you a few questions from the participants' end. Shall I ask you, sir?
Yeah, please.
Yeah. There is a question from Sony. Is AlphaFold limited to COVID-19 or can predict any protein structure using amino acid sequence? And if yes, then how can we use the AlphaFold?
Yes, actually, this AlphaFold is developed by a private company called DeepMind, which is funded by Google. Okay, so everything is not available right now, but people have tried to replicate the code and they are submitted in GitHub. So one can go to GitHub. GitHub is a repository for several softwares and codes. So there you can go and download a similar code, not exact code, but a similar code is available. So you can go and download that and you can try. So basically what I want to say is this AlphaFold is not really limited to COVID-19 target prediction. It can be applied to any protein. Okay, any protein. So if you know the sequence of the protein, AlphaFold can actually really get the fold of the protein. Okay. So that is possible. Okay, already it has shown its efficacy against 40 target proteins. Okay, so for example, in the last Cas13 conducted, AlphaFold was able to predict 25 protein structures with very, very high accuracy. Okay, so it matched the experimental results. So this took only a few days to predict. Okay, a matter of hours to predict. So that is the advantage. Whereas the existing other algorithms, okay, which are developed by other pharma companies, they were not able to match the AlphaFold's prediction accuracy. Okay, they were way far off. That is why AlphaFold was considered a disruptive technology. Okay, so now AlphaFold, as I want to say, AlphaFold is not restricted to COVID-19 targets. It can be applied to any target protein. Okay, so whose sequence is known? You need to get this, know the sequence of the protein. Once you know the sequence of the protein, the AlphaFold can actually, with very high accuracy, with reasonable accuracy, is able to get the fold of the protein, the structure of the protein. Okay. So, so the thing is, like, you don't need to know the identity. For example, in homology modeling also can predict, but we need to know the identity. At least 30% identity with a template protein. You need a template protein, which is sequentially very similar to your query protein, but whose structure is known. So, based on the known structure, you are trying to model the protein. That is what is homology modeling doing. But here in AlphaFold, you don't need any model. You don't need a homology model. So there are many situations where novel sequences are there, and we need to know their structures, predict the structures. In those situations, AlphaFold can be used. Okay. Yeah. Any other question?
Yeah. There is one more question.
Yeah. Please.
What is the scope after identifying the differentially expressed genes in human post-viral infection?
What is the
scope after identifying the differentially expressed genes in human post-viral infection?
Yeah, actually, like for example, now if you want to know like how this SARS actually is able to accomplish infection and how it's contributing the pathogenesis and mortality, in order to know that, we need to really find out the differential expression at the proteome level or at a transcriptome level. So identifying those will help us to know, for example, that the real reason why people die because of this COVID-19 is because of cytokine storming. Cytokines are produced in abnormal amounts, leading to various complications and death. So the thing is like how it's triggered, all those mechanisms can be elucidated. For example, how the virus enters. Now people say that it is because of this proteins like AC2 receptor and TMPSS2. Both these proteins are situated in the surface of the lung cells, and that is how the virus is able to attach with them and gets internalized by clathrin-mediated endocytosis. Okay, and once it is inside, what does it do? It tries to hijack the cell machinery so that it can make more and more of its own proteins. Okay, that means these this virus is able to somehow prevent the host cell from replicating its own protein. Rather, the host cell becomes a machinery to replicate the viral proteins. Okay, to make viral proteins, and it becomes a factory for the more and more virus to be formed. And this happens in a very short span of time. That means like in a very few days, the entire lung can be infected. Okay, that is why there's a higher mortality. Now, understanding at a proteome level or a transcriptomic level and getting the differential expression of the host genes, we'll be able to know which are the targets and how the virus is able to manipulate and cause infection, and how it's able to create this high mortality. We're able to be able to explain. Okay. Any other questions?
Yeah. Could you please let us know more about GPUs?
Yeah, this actually, for example, is a hardware requirement. It's a hardware requirement for running AI-based programs. For example, when you have a computer, you have CPU. Okay, so CPU means the RAM and all those things. Now, you have heard about graphic card in your computers. So nowadays, what happens is like these AI-based tools, like machine learning tools, they run on this GPU platform. Okay, so here GPU has several advantages. So combining CPU with GPU will actually lead to running those algorithms in a shorter span of time. Okay, so the thing is really you can accelerate the, for example, in a quicker time you can achieve that task. So nowadays, for example, you can have 12 GB RAM, GPU RAMs. I'm talking about graphical processing unit RAMs. The 12 GB RAMs, even people have got 24 GB RAM and also available. But the only thing is the price is a little higher. But anyway, now you have this Amazon Web Service, which can be used. You can always avail the computing platforms available in AWS, and you can you can get a domain there and you can use those computers. Okay, so the thing is that GPU is a hardware requirement for running advanced AI applications. Okay, so which cannot be done in a normal computer. It depends upon the algorithm. For example, there are very simple AI algorithms and there are very advanced AI algorithms. Okay, for example, the ones which are generally used in routine, for example, email check and all those email spam detection, bank fraud detection, those kind of things may use less sophisticated algorithms, but more sophisticated algorithms like, for example, self-driving and self-driving cars, those which are involved in protein structure prediction, all those may need a more sophisticated hardware. Okay, so they may need GPU. So if you have a GPU attached computer, it'll be good. Or you can, instead of wasting money on buying a computer, you can use the Amazon Web Service. You can go and rent out a computer there with all these features and pay them on hourly basis. Any other question?
Sir, thank you, sir. Thanks for your explanation.
Yeah.
As we are running out of time, we have disabled the chat option, sir. So that's the questions for this session.
Yeah,
Sir,
okay.
once again, we wholeheartedly thank you so much for your time and patience. Once again, thank you, sir. We look forward to get more lectures from your side. So yeah, thank you so much, sir. So participants, thanks for making this first day of e-workshop a great success. Thank you so much. See you all at 9:45 a.m. tomorrow. Thanks for your time and patience.